Our next speaker is Dr. Brian Fagan from University of Western Ontario. Dr. Fagan will give us an overview, clinical considerations and use of biosimilars for gastrointestinal disorders.
I abbreviated the title somewhat. Well, thank you, Gary. It's a pleasure to be here, and I think the intent of selecting me was to provide sort of a counterpoint to the regulatory opinion and try to put into perspective a clinician's view of a rapidly changing environment. And surprisingly enough, clinicians don't like change. And so the whole area is somewhat unsettling to them, and I'll try to go through why that is and perhaps give some reassurance, but maybe allow some also different, maybe new worries. So we've heard biosimilars are not generic drugs because they're not identical to the innovator. Why might it matter? Immunogenicity, and I'm going to come back to this theme. I mean, I think this is the crunch issue, really, from a clinical perspective. The immune system has developed over millions of years to do one thing, to differentiate self from non-self, and it's very good at it. It's exquisitely good at it. But it is influenced, whether you're sensitized or tolerized, by many covariates, and we have to think about those covariates in the clinical context. And sensitization has consequences, and gastroenterologists especially know this because when we first got our first biologic, within about a year we recognized that we had a huge problem with sensitization due to the way we were administering infliximab as a solo agent intermittently. That didn't turn out too really well. And so I guess I could cut to the chase here, and I think that the concept of interchangeability or non-medical switching really is the crunch issue from a clinical perspective, and I'll walk you through that. And little is known about that in humans. So we've heard earlier that these are complex molecules. They're not small molecules like aspirin or penicillin. And really one of the key issues is the quaternary structure, and that refers to the glycosylation patterns of monoclonal antibodies that are complex. And given the number of potential branching variations, the degrees of freedom in tacking on sugar molecules, there's an infinite number of structures with these molecules. Mammalian cells are bad employees. They don't do what you tell them to do. They do what they want to do when it comes to making sugar residues. So there's really a lot of complexity in structure and differences in identity. So I'm going to address just a couple issues, and I'll start with extrapolation and the whole approval process, which was addressed by Dr. Ritchie. And I think the issue here, there's a couple issues from a clinical perspective of why we feel that perhaps if we had a do-over again on the approval process for our first biosimilar, that we might do it a little bit differently. And I would submit that the regulatory environment has been influenced by the generic drug regulatory process. And why I say that is that there's two issues here. You know, the issue of comparability. The regulatory environment has requested not non-inferiority studies, but equivalency study. And that study is that concept is very important in generic drug development, where you're approving by PK. Because with small molecules, you want to know whether there's not enough drug or there's too much drug. So two-sided testing. Whereas in clinical medicine with monoclonal antibodies, generally a clinician just wants to know to make sure that the new product, the biosimilar product, is no worse than the reference product. And that has big sample size implications and estimation problems if you're trying to do two-sided testing and two-sided estimation. So that's one aspect of things that I think is a little bit different than the way we think in clinical medicine. Here's another one. This was the really pivotal study, Planteris, conducted in rheumatoid arthritis with an ACR 20 score as the endpoint. And you can see that it doesn't matter which bar is the active drug versus the innovator reference product versus the biosimilar. They're very similar for these endpoints at multiple time points. But one of the concepts that comes from a regulatory perspective is this notion of assay sensitivity. And you'll recognize the phrase there, assay sensitivity. Clinicians don't talk about assay sensitivity. So if I translate that, what that means is if a difference exists, you want the indication that is most sensitive to detect a difference clinically, if you're going to do a clinical study, that possible. You want the indication that is most specific and sensitive. And I would submit that that is not ACR20 scores in rheumatoid arthritis. The ACR20 score is a kind of fuzzy clinical measure. It is very nonspecific and high placebo rates associated with it. So what we might do that as an alternative, if I was asked a gastroenterologist which condition you would like to see a assay sensitivity highly specific study, it probably would be which of our diseases? Well, ulcerative colitis. We have a huge problem with pharmacokinetics in that situation, so there's high variability among patients, and it would really stress test a monoclonal antibody, unlike our ACR20 scores in rheumatoid arthritis. Moreover, we have a very objective endpoint in ulcerative colitis. We have endoscopy, which is highly sensitive and highly specific. So there's different philosophically, you can argue why this was done. Rheumatoid arthritis is 5 times as prevalent as ulcerative colitis, so it's easy to do the studies. So I'd suggest that there are philosophical differences between clinicians and regulatory authorities. No offense to regulatory authorities. So I think I can end this part of things. So the approval process, the extrapolation project, the game is over. The regulatory authorities are the highest court in the land, and globally they've ruled. And so these drugs are on the market, they've ruled safe and effective, and so substitution by your friendly pharmacist or payer is going to happen, and there's not a lot you're going to be able to do about that, and perhaps there's not a lot you should do about it, because they've met the bar that way. I would submit the far bigger problem is non-medical switching and the risk of immunogenicity. And we've learned the hard way that immunogenicity is a problem. And you can imagine multiple switching scenarios. So in the first case, it's a one-way switch. Then you can imagine multiple switches within a given product and reference product. And then very complex situation, we've already seen now, we have 2 TNF blocker biosimilars, or 3 actually. And you could imagine multiple switches with different products, and that gets very complicated. And perhaps this is an immunological stress test. We know in animal models that one of the ways of breaking tolerance is giving molecules that are very similar but not identical intermittently. So the question, if you're going to evaluate this, how many switches do you need to establish interchangeability? What clinical results are needed to be successful in establishing that? And then multiple different agents could be evaluated as well. The history of immunogenicity to foreign proteins goes back a long time, and this is Peter Medewar in Australia, who received, along with McFarlane and Burnett, the Nobel Prize in Medicine for their discovery of the factors that determine tolerance versus sensitization. And they did a number of experiments over the years working with influenza vaccine and worked this all out in the 1940s. And it's funny that we didn't really understand that in the 1990s when we started to approve foreign proteins for administration. But we've come to recapitulate the lessons that were available in the 40s. And essentially, there are multiple factors. Genetics are really important. You can breed animal strains, mice, that will never tolerize to a foreign protein. Alternatively, you can breed mice that will uniformly tolerize to a foreign protein. Route of administration is important, that generally if you put antigen into the skin subcutaneously, that's where the macrophages are in the tissue, and that tends to be a more immunogenic route than giving intravenously. Concomitant medications, we have numerous examples in gastroenterology where combination therapy is effective in preventing sensitization in distinction to monotherapy. How much drug you give matters, and I don't think I've ever seen a dose-finding study with any molecule in which the highest dose didn't have lower rates of sensitization than the lowest dose. And we used to blame that on that we didn't have drug-resistant assays. So if the old ELISA test that you can't detect antibody to drug in the presence of residual drug. And so, we said, well, that's probably why if you give more drug, you're less likely to detect anti-drug antibodies. We now have new generation ELISAs and HPLC tests that really get around that problem, and we're still seeing this phenomenon that high doses of drug actually reduce the risk of sensitization. And the most recent example, I think that is very relevant to gastroenterologists, is ustekinumab, which has recently been approved, and we're giving high doses, much higher doses than in psoriasis. And generally, we observe, when you compare broadly across diseases, psoriasis versus Crohn's disease, Crohn's intrinsically has higher rates of sensitization, and with the ustekinumab program, we're seeing lower rates than we observed with psoriasis with the same assay. And this may be a reflection that we're giving a lot more drug up front and tending to tolerize. One really important lesson we've learned, that it's not the cell line that you grow the protein up that determines immunogenicity. There are no human amino acids. There's just the three-dimensional structure of the foreign protein and your T cell repertoire, and those two interact together to determine whether you sensitize or tolerize. So let's talk about the experience in IBD. Here's our original first look at this problem in the New England Journal. This came out of Leuven, and in the days of intermittent use of infliximab, which was a really bad strategy, we saw that the anti-drug antibody titer determined the time in remission. There was a positive association in that if you had anti-drug antibodies, that you were less likely to be in remission and less likely to stay duration of remission. And that also was associated with the presence of infusion reactions. Just to reinforce the concept that humanization does not solve this problem, this is a very large cohort in rheumatoid arthritis in the Netherlands, looking at patients from new onset treated with adalimumab and looking at the proportion of anti-drug antibodies to adalimumab over time. And this population had a high prevalence of background methotrexate therapy as well. And what you see here is that you get anti-drug antibody formation rates of approaching 30%, and that the majority of sensitization occurs within the first 12 weeks or so, with a molecule that is, quotation marks, fully human. So you can imagine that if we're going to have drugs that aren't the same, and we're switching between them, that we might have a problem with immunogenicity as a stress test. And so we need to evaluate that with potential switch studies. And I understand the FDA has recently issued a guidance about the types of studies that they might want to see to allow non-medical switching in the context of having multiple different agents available. And perhaps we can talk about that later. One thing I will say is that I don't think this study has helped very much. This is the NorSwitch study. It was funded by the Norwegian government, and it was quite amusing when the investigator presented this study for the first time. He said, one real advantage of this study is it's supported by the Norwegian government, so we're not biased like those pharma studies. So I mean, who's going to be the benefit of getting biosimilars? It's going to be the Norwegian government, but I'll give them that. The Norwegian government did fund this study to their credit. And this was a one-way switch study with CTP13, which is Inflectra, versus infliximab, reference product. And essentially the patients had stable remission for 6 months, and then they were randomized 1-to-1 to switch to the drug, and they had an open label extension out to 78 weeks, but the primary endpoint was at week 52. Now, Norway's a small country, and to actually do the study, they took the expedient route of entering multiple diseases. And immediately, this alienated a lot of clinicians. However, having said that, 50% of the patients in the trial either had Crohn's disease or ulcerative colitis. So you had to be in stable remission, and obviously you had to have definitions of remission that were different across the different indications. And then similarly, for the primary endpoint, the endpoint was failure, disease worsening, and that would have to be unique to all of the different indications. Now, here's an issue that, as a methodologist, a problem. The disease margin here was 15%, and I'm not going to terrorize you with all the issues of non-inferiority study, but this is a problem because certainly in IBD, we would consider a non-inferiority margin of 15% as very generous, and I think that equally applies to rheumatoid arthritis. A non-inferiority margin in IBD, we would consider a minimum clinically important difference of 15% as being a minimum clinically important difference. So you can use a very empiric rule. If you cut the clinically important difference in half, a lot of times that translates into a non-important difference, and that would be 7.5%. If you do the sample size for that calculation, you end up with a number of around 1,200 patients, and this was far less than that. just illustrating that there was a good proportion of Crohn's disease and ulcerative colitis patients in this trial. And here's the primary analysis. If you turn down to the point estimate, and these are presented as two-sided confidence intervals, you can see that the line of identity overlaps 1, so this is a positive study. The point estimate favored the reference product, but by the definition of the primary endpoint with a 15% margin, this study met its primary endpoint. Clinicians love subgroup analyses, and so when they saw these data, the first question is, well, what about the subgroup analysis in my disease? And one can see that with Crohn's disease, the point estimate actually comes very close to the line of identity, indicating, you see down at the bottom here, I don't have the pointer, and this immediately set off a furor that, in fact, this reinforces my bias that Crohn's disease is different in rheumatoid arthritis, and I can't accept these results. I don't think that's a very valid criticism of this study. It wasn't powered to show differences in subgroup analysis. And in fact, if you actually look at a variety of other endpoints with regard to Crohn's disease and the other indications, there really isn't a compelling, there's not consistency of the data, which would reinforce that the subgroup conclusion is valid. If you saw the same thing with multiple endpoints, you might accept that maybe it is real. But this should be hypothesis generating, not concluding that NOR switch did not show a benefit, well, non-inferiority, oh God, in Crohn's disease. So I'm getting near the end here. And okay, I'm not going to go back and do that. So one of the slides I showed you was a snowflake. Okay, the last slide I had was a snowflake, and it showed different snowflakes, and the old adage is that no two snowflakes are alike. And this is where I think the whole crunch question comes down to. So if you accept the fact that there's molecular heterogeneity within the reference drug, then what I said about being exposed to different molecules intermittently, that experiment has already been done. And that's why you have a 10% rate of sensitization with monotherapy with the monoclonals. So don't worry about all this non-medical switching. It's all going to be fine because you've already been exposed to heterogeneous mixes of molecules. I think that's a hypothesis, and it needs to be tested. And at this, as someone who's interested in immunogenicity and epidemiology and safety of patients, I would really like to see an experiment before I condone non-medical switching. And I think that at least the regulatory authority in the United States and Canada, I think, are coming around to that viewpoint, but I'd be interested in what the most recent perspective is. So to summarize, the reality is that the biosimilars are here. There are going to be substitution based on cost. The only advantage of them really is cost. And we're going to see that, and you're going to have to live with that. What's going to happen with non-medical switching remains to be seen, and I think that's something we should, in the absence of data, I'm not going to condone that personally as a physician. So I'll stop there.
Thank you.